last time we have discussed the glumerular anatomy and glumerular filtration rate. Also, we have discussed the pathological conditions like anti-GBM antibodies, Goods posture syndrome, minimal change glomerulitis, etc. Today we will discuss the proximal convoluted tubules, which is the second part of nephron. Generally, nephron have how many parts? For example, I have generally a nephron. Nephron is divided into two parts. One is renal corpuscle and one is renal tubules. Renal corpuscle is composed of glumerulus and Bowman's capsule. These renal tubules are proximal convoluted tubules, loop of Henle, distal convoluted tubules, and collecting ducts. Right? We have discussed these two parts, the glumerulus and Bowman's capsule. The glumerular epithelium, which is composed of podocytes, or the capillary stuff, which is composed of fenestrated epithelium, which is responsible for filtration. And the basement membrane is the other barrier between the Bowman's capsule and glomerulus. And the nebulin proteins, which are present in between the podocytes, is another barrier for filtration. How many things were filtered from glomerulus? For example, I have this efferent arteriole that is divided into a lot of capillaries and forms the glomerulus. Glomerulus is placed in a cup-like structure called Bowman's capsule. And after we have proximal convoluted tubules, then we have loop of Henle, we have distal convoluted tubules, and at last we have collecting ducts. Right? This is glomerulus, this is Bowman's capsule, this is proximal convoluted tubules, this is loop of Henle, this is distal convoluted tubules, and these are collecting ducts. Also, we know that this efferent arteriole is again divided into capillaries, and these capillaries are running with renal tubules. Today in this lecture we will focus on this proximal convoluted tubules. What are absorbing and what are secreting by blood to these tubules, because we know that in glomerulus water is filtered. Electrolytes like chloride ion, sodium ion, glucose, magnesium ions, etc. Organic nutrients like glucose, amino acids, etc. They are all here filtered. Now we will take just this part from this nephron and we'll see what processes occurs in proximal convoluted tubules. These processes are very important in urine formation. Right? After discussing the proximal convoluted tubules, loop of Henle, distal convoluted tubules, we will go for urine formation. And after studying all these parts, urine formation is very simple. Right? So we are taking the proximal convoluted tubules. For example, this is the efferent. This is the efferent arteriole. This is the glomerulus, for example, and this is the efferent arteriole. right? The blood is coming to glomerulus from efferent artery. This thing is called glumerulus, and this is efferent artery, right? This structure is placed in a cup-like structure called glumerulus, and glumerulus is like a funnel. Here Bowman's capsule is like a funnel. Now for example, this is the proximal convoluted tubule. And this is for example loop of Henle. We are discussing only proximal convoluted tubules. So we are taking only proximal convoluted tubules. The important point, which things are coming to proximal convoluted from Bowman's capsule. This is important because all the filtrate, all the things which are coming, which are filtered from glomerulus to Bowman's capsule were reabsorbed from this proximal convoluted tubules. For example, this artery, this blood supply is running between or near to this renal tubule. So for example, this is the blood, and of course blood capillaries are formed of simple squamous epithelium. Simple squamous epithelium. Now the question is, which type of epithelium is present in this tubule? Right? This proximal convoluted tubule, for example, proximal convoluted tubules is composed of cuboidal epithelium. Which type of epithelium is present in proximal convoluted tubules? Cuboidal epithelium. But this is special type of cuboidal because on lumen side, this is the lumen, this is the inside of tubule. There are some finger-like projections on this cuboidal epithelium. Like for example, this is Bowman's capsule, and this is proximal convoluted tubule. This is loop of Henle. Here the epithelium have a brush borders. They have brush borders. First thing is this, it have brush borders on lumen side. Lumen side. Second, it is highly permeable to water and electrolytes, right? The proximal convoluted tubule cells are highly permeable to water and electrolytes, mean that the water and electrolytes are easily passed from proximal convoluted tubules, and this is the reason why most of reabsorption occur in proximal convoluted tubules. There are some special type of transporters present in this epithelium. So these are the special characters of cuboidal epithelium which are present in the proximal convoluted tubules at receiving the things from Bowman's capsules, and Bowman's capsule receiving the things from glomerulus after filtration, right? So for example, this is the blood, and of course here is the basement. What is the name of this basement membrane? GBM. This is called GBM, glomerular basement membrane. Glumerular basement membrane. One other important point here is, for example, this is a kidney. This is kidney, and in kidney we have cortex and medulla, pyramids. Here are pyramids. For example, I am taking this nephron, which are we going to discuss. As for example, here The Bowman's capsule and the glomerulus are present in the cortex. Right? They are present in cortex. The proximal convoluted tubules also present in cortex. But the loop of Henle of this type of neurons are present in medulla. And remember, medulla is very concentrated in ions like electrolytes, chloride ions, sodium ions, magnesium ions, potassium ions. They are very concentrated in medulla, but they are not concentrated in the cortex. And this is the reason why we can easily reabsorb and absorb these electrolytes and proximal convoluted tubules, because proximal convoluted tubules are present in cortex. Right? From here, this is blood. From blood, it takes water. This is the first thing. During glumerular filtration, this Bowman's capsule receiving water. Second, it receiving electrolytes. Electrolytes like the first one is sodium, chloride, bicarbonates, HCO3 minus, magnesium, potassium, calcium, etc. All these electrolytes are coming from blood into Bowman's capsule. All the organic nutrients mean organic nutrients like glucose, lipids, vitamins, and amino acids. They are coming to Bowman's capsule, right? Some of the very small proteins, very small proteins, because I have told you that protein cannot cross this all these membranes like capillaries wall, the basement membranes, and Bowman's capsule wall. But very small protein like insulin can cross these very small proteins, can cross this glumerulus. And the example of this is, for example, insulin coming from pancreas. These are the things which are coming from the capillaries into Bowman's capsule during glumerular filtration. What will be the osmolality of this blood? Osmolality, I have already discussed this. 300 milliosmol per liter. What is the net filtration pressure? Net filtration pressure is 10 mm of Hg, right? 10 mm of Hg. Under all these hydrostatic pressures, these nutrients filter from the blood into Bowman's capsule, and this step is called glumerular filtration. Glumerular filtration. Remember that in urine formation, we have three steps. First is glumerular filtration. This is glumerular filtration. Second thing is reabsorption. We will discuss reabsorption here, right? And the third is tubular secretions. We will discuss these absorption, secretions, and reabsorption in all the parts of nephron. The first part is proximal convoluted tubule. So let's, for example, this proximal convoluted tubules have epithelium and having brush borders like this. One other character of this proximal convoluted tubules cuboidal epithelium is this epithelium is very rich in mitochondria. Why? Because all the transports occurs here will need energy, ATP, because the electrolytes are transporting against the concentration gradient, right? So it will require energy. So the cuboidal epithelium of the proximal convoluted tubules is rich in mitochondria, right? This is one cell, for example. This is other cell. This is the next cell, and here we have, for example, this is one cell and this is other cell. These cells are called the cuboidal epithelium. One other special character is there are some gaps between the cells of proximal convoluted tubule, and these gaps are very important for transports, absorption and reabsorption, right? So proximal convoluted tubules receives all these things from glumerular filtration. We will discuss, first of all, we will discuss the reabsorption of glucose. Secondly, we will discuss the reabsorption of amino acids. Third, we will discuss the reabsorption of lactic acids. We will discuss the reabsorption of lipids. Then we will discuss the reabsorption of water and sodium. Then we will discuss the chloride, potassium, magnesium and calcium reabsorption. And in last we will discuss what is the effect of parathyroid hormones on proximal convoluted tubules, because this parathyroid hormone is responsible for electrolyte balance in body, right? And in last we will discuss the secretions in proximal convoluted tubule. First we will discuss all the reabsorption. Reabsorption, what is the difference between reabsorption and secretion? Anyone? What is the difference between the reabsorption and secretion? Secretion? The blood na chikam proximal convoluted tubule. Absorption chikala the tubules na blood taraz. This is good. For example, this is proximal convoluted tubule. This is other proximal convoluted tubule, and this is blood. When the things coming from the tubules, from tubule into blood, this is called reabsorption, right? And when the things are coming from blood into the tubes, it is called tubular secretion. Clear? This is the secretion. So first we will discuss the mechanisms of reabsorption, and then we will go for secretion, because there are only two secretions occur in proximal convoluted tubule, but the reabsorption is maximum in proximal convoluted tubule as compared to all other nephron, right? First of all, for example, this is the blood. This is the blood supply, right? These are the interstitial spaces. These are the interstitial spaces. So from here, sodium is present in cell, right? And potassium is present here. See theory, the sodium occurs maximum in interstitial fluids and potassium maximum present in inside the cell, right? Here we have a transporter in all these cells. Here, for example, these red transporters are called potassium sodium co-transporters. They are called potassium sodium or sodium potassium co-transporters. They are called sodium potassium co-transporter. Anyone can tell me what is the difference between primary active transport and secondary active transport? Because we will need it. Primary active transport or active diffusion and secondary active diffusion. Let's see. For example, this is cell, right? And here the sodium level is high, and here the sodium level is low. And this sodium is trying to go from low concentration into high concentration with the use of ATP. This is called primary active transport. Primary active transport. But for example, we have a cell here, and this is a transporter here, and we have glucose outside, and we have sodium inside. Here the sodium concentration is high, and here the glucose concentration is high. Both these contents are trying to go against concentration gradient. For example, the sodium is trying to go from lower concentration into high concentration, right? And this glucose is also going from low concentration into high concentration. This is active diffusion, but here it doesn't use ATP, because with the sodium, this glucose follows the pathway and enters into the cell against the concentration gradient through the glucose, through the sodium. So this is called secondary active transport. So let's see here. For example, inside the cell the sodium content is low, and outside sodium is in high concentration, right? And inside the potassium concentration is high, and outside the potassium concentration is low. This is general, right? Here three sodiums will go from inside into outside, mean that it's going against concentration gradient, right? The potassium, when three sodium going outside, two potassium will enter. For example, three sodiums go outside, two potassium will go inside. This is a very active MCQ in your paper, that for 3 sodium export, how many potassium are required? For 3 it's required 2 potassium, and 2 potassium required 3 sodiums, right? Now for example, from here sodium is going outside, and potassium is going inside. The sodium concentration inside the cell decreasing, right? So for example, here we have sodium, here the sodium concentration is high, and inside the cell the sodium concentration is decreasing. In the proximal convoluted tubule cells, we have a special co-transporter here, and this transporter is having a very special name. It is called the sodium glucose co-transporter, and it follows the secondary active transport, because when we are taking nutrients, the glucose is already high in cells, but glucose here is very low as compared to inside the cell. This sodium is going against concentration gradient, or in concentration gradient. That is going in concentration gradient, because sodium is outing from inside the cell, so it makes a concentration gradient here. But this sodium, it hold the hand of glucose, and with sodium, glucose is also going in cell against concentration gradient. So this glucose is also going to cell, but this glucose is going against concentration gradient. So this transport is called secondary actio transport. This is called secondary actio transport. Right? And here for glucose we have GLUT receptors. Remember, GLUT receptor, GLUT1, GLUT2, GLUT3, GLUT4. Right? So here we have GLUT receptor, these are called glucose receptor, and by this glucose receptor, this glucose leaves the cell of the proximal convoluted tubules and reabsorbed into blood. This is the first reabsorption of glucose. The mechanism is secondary actio transport. The electrolyte required for the reabsorption of glucose is sodium. Right? Here, this glucose in proximal convoluted tubules, 100% of glucose is reabsorbed. This value is very important. 100% of glucose is reabsorbed. For example, 100 glucose coming from Glumerolus into Bowman's capsule, from proximal convoluted tubules, that 100 by 100 glucose is going into blood again. Right? The next thing is we have amino acids. For example, this is amino acid. Different types of amino acid. We have 21 types of different amino acids, and some of the amino acids can cross these membrane barriers and come into proximal convoluted tubules. From proximal convoluted tubules, we have a transporter here. This transporter is called the sodium glucose co-transporter. This transporter is called amino acid, sodium amino acid co-transporter, and the same mechanism is followed by this transporter. This sodium is going in concentration gradient with this sodium. This amino acid is also going into cell, and here we have a special types of transporter called amino acid protein transporter. It takes amino acids from here into the blood, amino acids, and also 100% of amino acids are reabsorbed in proximal convoluted tubules. If you found in MCQ, for example, how many amino acids are reabsorbed in distal convoluted tubules? 100%, 90%, 80%, 0%. 0%, because all the amino acids are reabsorbed in proximal convoluted tubules, not in distal convoluted tubule. Right? This sodium also leaves the cell and goes into blood, right? And some of the sodium came into here, and some of the sodium, for example, from 3 sodium from here goes into the interstitial fluids, and it takes 2 potassium into cell, right? Now here this is the reabsorption of amino acids. 100% of amino acids are reabsorbed. Next, we have lactic acids, because during fermentation lactic acids are also formed. For example, we are exercising, lactic acids are forming in muscle cell, and they are adding into blood, and the blood came into Glumerolus, and from Glumerolus the lactic acid is reabsorbed. Lactic acid also reabsorbed with this glucose, lactic acids, lactates. It follows the glucose transporter and goes into blood, and lactic acid is also absorbed 100% here. Lactic acid also reabsorbed 100% in proximal convoluted tubule. Next is, what will happen to the lipids? The lipids mechanism is different, because lipids is lipophilic. It can dissolve in the phospholipids. So for example, here we have lipids. This is lipid. But all the other thing like lipids, for example urea, it will attach to this lipid and will goes into cell, and from here it will goes into the blood, and 100% of lipids are reabsorbed in proximal convoluted tubule. This is the pathway, and this is called the transcellular transport. This is called secondary active transport, and this is called the transcellular transport mechanism. What is transcellular? When it is going from the cell membrane into cytoplasm, and from cytoplasm it goes into the blood. For example, this is the cell, and for example lipids are going from here up to here. This is called the transcellular transport. So lipids leaves the lumen of the proximal convoluted tubules by transcellular transport, right? Glucose, amino acids, they are, and lactic acid, they are leaving the lumen of the proximal convoluted tubules by secondary active transport. These sodium and potassium, sodium leaves the cell proximal convoluted tubules through primary active transport. Because it is going against concentration gradient, it will use energy. It will use ATP into ADP, right? And these are called the sodium potassium ATPase transporters. Sodium potassium ATPase transporters, right? This is called the primary active transport. It is called primary active transport, because it is consuming the energy directly, right? This is the lipids transport, which is transcellular transport. Next, we also have these electrolytes. The electrolytes, for example, we are taking chloride. Here we have blood. This is, for example, blood capillary. Blood capillaries. We are taking chloride. Two mechanisms are followed by this chloride ion. One is paracellular transport. One mechanism is the sodium chloride co-transporter mechanism, right? For example, this chloride ion goes in between the cuboidal epithelium of proximal convoluted tubules, and from here we have a special transporter called the chloride transporter. It takes the chloride into blood, but against this chloride ions we will see that bicarbonate ions will enter into blood, right? We will discuss this last. But this mechanism is called the paracellular transport, because this transport occur in between the two epithelium, right? Paracellular transport. Paracellular transport mechanism. This is the fourth one. The first one is primary active, the second one is secondary active, the third one is transcellular, the fourth one is paracellular transport. And the other 55% of chloride is reabsorbed in proximal convoluted tubules. How many? 55%. 55% chloride reabsorbed in proximal convoluted tubules. The magnesium, calcium and potassium. These electrolytes also follows the paracellular transport and came it into blood. Potassium, magnesium and calcium, and it follows the paracellular transport. The second mechanism for this chloride ion is, for example, we have a transport here, and this transporter is called chloride sodium co-transporter, right? Here we have chloride ion and we also have sodium. Here we also have sodium, and so this chloride will go from here, and this sodium is also go from here through the chloride channel. This chloride will goes into the blood, and this sodium will go into interstitial fluid. This is the next mechanism, which is called the simple diffusion. Simple diffusion, this is paracellular transport, this is transcellular transport, this is secondary reactive transport and primary reactive transport, right? The next thing, which one is remaining? The very small proteins like insulin, because when a lot of proteins are filtering from the glomerulus, is called proteinuria, and this is a condition in which we can diagnose that our kidneys are not working properly. But here we have also a very small proteins like insulin, and insulins can easily filter from the glomerulus. And here the next mechanism is endocytosis. For example, remember recap the receptors I have discussed in biochemistry. One pass receptors, insulin receptor, tyrosine kinase receptor. For example, here I have one pass receptor, special type of receptor for insulin, and this is, for example, insulin. This is insulin. This insulin came and binds up to this receptor, and from here it starts the endocytosis, and in endocytosis it encloses the receptor as well as the insulin. And we are well aware that receptors are also made up of proteins, and this insulin is also protein. Here this is now lysosome. This is lysosome. Our lysosome came and secrete the hydrolytic or lysosomal enzymes on these proteins and convert all these protein into amino acids, and by exocytosis, like for example, these amino acids are reabsorbed into blood. Amino acids made from insulin. For example, if we have some glomerular dysfunctions or glomerular issues, and hemoglobin is also filtering from this glomerulus. Here in these receptors, for example, one will be insulin and the second will be hemoglobin, right? And the same mechanism will follow by these lysosome. It will break the hemoglobin and the insulin into amino acids, and these amino acids will be reabsorbed into blood. This is the next mechanism, and this mechanism is called endocytosis mechanism. Endocytosis mechanism, right? Insulins and other things follow these mechanism, right? So these are the reabsorption of certain molecules from proximal convoluted tubules, right? The next, water and sodium. Remember one thing, in all over the body, water follows sodium and sodium follows water. When water is going from one compartment to other compartment, sodium will also go from that compartment into other. And when the sodium is trying to leave one compartment of body and goes into other compartment, the water must leave that compartment because sodium is dissolved in water, right? So for example, now we are discussing the reabsorption of water. For example, I have cells from proximal convoluted tubules. These are proximal convoluted tubules, right? And we have water here because a lot of water is filtering from glumerulus, right? From here the water will go into the cell, and from cell it will go into the blood. This is the reabsorption of water. This is the one mechanism which is not occurring in the ascending limb of loop of Henle, right? But I have told you that sodium will also follow the water. So sodium will follow water and goes into the blood. How many blood is reabsorbed in proximal convoluted tubules? 65%. 65%, right? And sodium is also reabsorbed 65% in proximal convoluted tubules. These values are very important for MCQ, right? Write it down with yourself. H2 water is reabsorbed from proximal convoluted tubules into blood is 65%, and sodium is also 65%, right? So this is the reabsorption of water and sodium, right? First we have discussed the reabsorption of glucose, then the reabsorption of amino acid, reabsorption of lactic acids, reabsorption of chloride ions, reabsorption of magnesium, potassium and calcium ions, reabsorption of insulin, then reabsorption of water and sodium in proximal convoluted tubule, right? Now we are going for the secretions. Which things will leave the blood and will goes into the lumen? Because for example, if we are eating or some reactions taking place in during metabolism, and it causes the pH of blood lower from 7.4, and we called it metabolic acidosis. But in kidneys we have a mechanism, very active mechanism, which maintain the pH of blood. What is that mechanism? For example, if the pH of the blood is lowering down, the pH of the blood is normal pH of blood is 7.35 to 7.45. This is the normal pH of blood. For example, if we are caused by metabolic acidosis, for the pH of blood is less than 7.35, right? Here we have a remaining one electrolyte which is called the bicarbonate ion, and this bicarbonate ion is very important for maintaining the pH of the blood, right? But it cannot cross the membranes directly and goes into blood. So what will happen? In blood we have carbon dioxide, right? And the water is reabsorbing from the proximal convoluted tubules and blood. Here it will react with water and it will form a third compound which is called carbonic acid. It's called carbonic acid. But this carbonic acid is very unstable compound. Very unstable, right? So from here it will again break down. Right, we will come from to this mechanism, but what happens to here to the bicarbonate ion, right? The bicarbonate ion here, it is present in HCO3 minus. HCO3, this is the bicarbonate ion. This bicarbonate ion will react with which thing? With hydrogen ion. It will react with hydrogen ion. This hydrogen, where is this hydrogen ion coming from? This hydrogen ion is coming from here. It will break down into H plus and HCO3 minus. Right? This hydrogen ion will reabsorb into, will secrete it into the proximal convoluted tubule epithelium, and from here it will go into the tubules, right? And when these two things will react, it will form H2CO3, bicarbonate and carbonic acid, and this carbonic acid here will break down into these two compounds, right? The CO2 and H2O, right? And here this bicarbonate acid, which enzyme is responsible for catalyzing this reaction? Carbonic anhydrase. Carbonic anhydrase enzyme is responsible for catalyzing this reaction. This carbonic acid is very unstable. It will break it into hydrogen ion and HCO3 minus. HCO3 minus is responsible for increasing the pH of blood, and in metabolic acidosis the pH of blood decreases. This bicarbonate ion is responsible for increasing the pH of the blood, so it prevents the metabolic acidosis, and this hydrogen ion is secreted into the tubules, proximal convoluted tubules. Here this bicarbonate ion is coming from glumerulus. From glumerulus, this bicarbonate ion will react with hydrogen which is coming from the blood into proximal convoluted tubule, and will form the carbonic acid. This reaction is also catalyzing by carbonic anhydrase, and this carbonic anhydrase are very unstable, so it will break down into the carbon dioxide and water, and these two will be secreted in urine, right? Next we have also phosphates here, HPO4 minus, HPO4 minus, right? Here we have a very special transporter which is called the phosphate sodium co-transporter. We have sodium also, and this sodium is coming HPO4 minus, but if this phosphate or phosphoric acid will adding into the blood, it will increase the pH of the blood and will go the blood into alkalosis, because it increasing the pH of blood. So for preventing, we have a special type of receptor on these cells which are 7-pass receptors. 7-pass receptor, also known as serpentine receptor, and also known as G protein coupled receptor. Here in blood, for example, from thyroid, this is the thyroid gland, and in thyroid gland we have 4 very small glands called parathyroid glands, and parathyroid hormones are secreted from parathyroid glands. These parathyroid hormones are adding into the blood, and it will came into here, and the parathyroid hormone will attach with these receptors which are GS receptors, stimulatory receptors. This is one of the exceptional case in body, right? When this receptor is activated, it will follow the pathway, because it will activate the adenyl cyclase, and adenyl cyclase will, for example, here we have alpha, beta and gamma proteins. These alpha, beta and gamma protein will activate adenyl cyclase. These adenyl cyclase will activate the ATP N2C-AMP. This CMP will activate the phosphokinase A, right? Phosphokinase A is responsible for phosphorylating the channels, right? We all are thinking that when this channel is phosphorylate, it will stimulate it, but this is not like that stimulatory mechanism. By phosphorylating this transporter, it inhibited, and this phosphoric acid is not able to go into blood, and it will prevent the alkalosis in body. The blood alkalosis will be prevented by this receptor. So the blood alkalosis is protected by the parathyroid hormone by activating the GS receptor in the proximal convoluted tubules to prevent the reabsorption of phosphoric acid into blood, right? This is the next thing. The next is, for example, we have bile salts. We have bile salts in blood which are coming from liver. We have drugs, especially the antibiotics, penicillin, the cephalosporins antibiotics. They all are filtrating from glumerulus, right? For example, some of the oxalate ions present in blood, they all are secreted into the proximal convoluted tubules. Biles, drugs, and oxalate ions, etc., and will go and excrete from the body through urine, right? Another mechanism for preventing the acidity of the blood is the breakdown of glutamines. The reactions occur is called deamination. In deamination, it will produce two amine groups and one bicarbonate ion, and bicarbonate ion will go into the blood and will prevent the decreasing of the pH of blood. So this is all the mechanism of occurring in the proximal convoluted tubule. Suppose after this we will discuss the distal convoluted, we will discuss the loop of Henle in detail, and after loop of Henle we will divide the distal convoluted tubules into two parts, the early distal and late distal, and we will discuss all the mechanisms occur in that distal convoluted tubule. Here the normal values are, what are reabsorption values? Reabsorption, tubular reabsorption. Water is reabsorbed 65% in proximal convoluted tubules. Sodium is 65%, right? Chloride ions 55%, magnesium ions 40%, potassium and calcium also, right? Glucose 100%, lipids 100%, small proteins 100%. Not all the vitamins are reabsorbed in proximal convoluted tubules, right? Some of the vitamins are reabsorbed in proximal convoluted tubules, only the vitamins which are water soluble, right? So this is the mechanisms occur in proximal convoluted tubule. The first mechanism is primary active transport, and by primary active transport the sodium leaves the cell. The sodium leaves this cell and goes into interstitial fluids, and the potassium leaves the interstitial fluids and goes into the cell, and this is against the concentration gradient, and it is using ATP, so it is called primary active transport. The second mechanism is the reabsorption of glucose. This reabsorption occur against concentration gradient but occur with the help of other electrolytes, so it is called secondary active transport. The next is the lipids and urea, which following the transcellular transport. The next is the chloride ions, the magnesium, calcium and potassium, they are following the paracellular transport. Paracellular transport means when the things are transporting in between the two epithelium. And the last one is the endocytosis, and endocytosis, the whole proteins or the whole thing is enclosed in the vesicle, and lysosomal enzymes work upon that proteins and other macromolecules and convert it into small fragments and reabsorbed into the blood, and the blood takes these amino acids into the liver, and liver is responsible for forming other substances from amino acids. The next is the secretions, the reabsorption of water. This reabsorption of water follows the sodium, and both the water and sodiums are 65% reabsorbed in proximal convoluted tubules. And last we have secretion, the hydrogen ion, the carbon dioxide and the bicarbonate ions, and directly they secrete into the renal tubules. And in last we have bile salts, drugs and oxalate ions are reabsorbed into, are secreted into the proximal convoluted tubules. After proximal convoluted tubules, all the things which are present in the proximal convoluted tubules will go into the loop of Henle, and we will see that which things will reabsorb or secrete in loop of Henle, right? If someone asks that what is proximal convoluted tubule, proximal convoluted tubule is a very important part of nephron, which is present just after the Bowman's capsule, or which is directly attached with the Bowman's capsule. It's called proximal convoluted tubule, or most of the reabsorptions occur in the proximal convoluted parts of nephron, right? So कोई नहीं